Hi everyone, it's Dr. LeBlanc with Unit 7 on mutagenesis and repair. Our first lecture in this sequence is on DNA sequencing errors. Our learning goals are to understand that mutations provide the basis for evolutionary change, to understand that most mutations are deleterious, to understand that many different ways to repair and overcome mutations have evolved, to understand the difference between wild-type, mutant, and reversion mutations, to understand that changing the sequence of nucleotides in a gene will alter its function, to understand that mutations in the regulatory and coding regions of a gene can alter its function, and to understand that suppressor mutations can overcome some mutations. There are three big picture ideas about mutagenesis and repair. First of all, mutations provide allelic variations. They are the foundations for evolutionary change. However, most mutations that occur within an individual or found in a population are often detrimental. Mutations are heritable changes in the genetic material, and repair mechanisms are many and overlapping. Let's get a few terms straight about mutations and their effects on genotype and phenotype. First of all, wild type is the prevalent genotype. That is the phenotype, the genotype that corresponds to the phenotype that you can observe most frequently within a population. A forward mutation changes the wild type into a new variant or a mutant, and a reverse mutation changes the mutant allele back to the wild type. That is what a revertant means, that you've changed a mutant back to the wild type. A point mutation is a change in a single base pair. It involves a base substitution. Here we have a sequence of nucleotides, and we're going to take a look specifically at this T right here. When we change a nucleotide to a different nucleotide, that's a point mutation. Transitions are changing a pyrimidine to another pyrimidine or a purine to another purine. Transversions are changes from a pyrimidine to a purine or vice versa, or purine back to a pyrimidine. Transitions, these are much more common than transversions. Not so common. Okay, let's talk for a little bit about mutations that occur within the coding regions of genes, in the regions of the genes that encode amino acids. Okay, they would occur within exons. Here's our basic sequence right here. We start with the methionine. And remember that every 3 nucleotides encodes an amino acid. A silent mutation would change a nucleotide from a G to a C without any changes in the amino acid sequence. So CCG and CCC both encode proline. Remember, the last nucleotide of a codon is often referred to as the wobble base. So frequently, if you change the 3rd nucleotide of a codon, you're not going to change the amino acid that is encoded by that codon. What about a missense mutation? Here we're changing this ACC, to a CCC. Well, ACC encodes threonine, but CCC encodes proline. So a missense mutation changes the amino acid. Okay, that often has a consequence for protein function. Sometimes it doesn't affect it at all or detectably. Other times it can have very profound difference in their protein function. For example, in sickle cell anemia results from a single base pair substitution in one codon, one amino acid. This is what normal red blood cells look like. They look like little frisbees. When you change that single nucleotide and a single amino acid, you end up with these misshapen red blood cells. This is the substitution that occurs. Normally, the Sixth amino acid encodes is glutamic acid. That's the normal amino acid in this hemoglobin. However, if you change this T to an A, you change this codon from CTC to CAC, and that encodes valine instead of glutamic acid. What does that do for the red blood cells? Well, the change in amino acid sequence causes the hemoglobin molecule to crystallize when oxygen levels in the blood are low. As a consequence, red blood cells sickle and get stuck in the small blood vessels. Sickle cell anemia evolved as an adaptation in humans against the malarial parasite, Plasmodium. Well, there's a bunch of different plasmodia that cause malaria in humans. If an individual is heterozygous for this trait, in other words, they have one good copy and one mutant copy, they are less susceptible to infection by the malarial parasite. Of course, if they're homozygous, then they end up with the sickle cell disease. So there's a trade-off. If you have a nonsense mutation, you change an amino acid codon into a stop codon. TAA is a stop codon, and so this polypeptide ends up only being 4 amino acids long. There's a definite change in protein function. It is non-functional. And most of the time, a tiny little peptide like this will be quickly degraded in the cell. Can also have frame shift mutations, addition or deletions of nucleotides. That will change not only the codon in which that nucleotide change occurs, but also all subsequent codons. So you completely change the amino acid sequence after that point. Not only do you change the amino acid sequence, but you also usually introduce early stop codons. So there's a definite change in protein function that occurs as a result of frame shift mutations. So let's look a little bit more closely at a particular frame shift. Anytime you have an insertion or deletion that is anything other than a multiple of 3, you will end up changing not only that amino acid where the insertion or deletion occurs, but all of the subsequent amino acids that are translated from that messenger RNA will be different than from the original. So if you have a 4-base pair deletion, here we have a nucleotide sequence. We're going to remove these 4 nucleotides right here. Okay, so now this CTC is right up against this G here. So we've changed the codon. This is the original amino acid sequence, asparagine, glutamine, isoleucine. But because we took out 4 nucleotides, not only will change this glutamine, but we'll also change any subsequent amino acids afterwards. So we've changed the glutamine to alanine, and that isoleucine is no longer encoded. The sequence is going to be different. What if you insert a multiple, anything other than a multiple of 3? So at this point, we're going to insert some nucleotides right here between this C and this G, between this C and this G. So we've altered this codon. Now we're going to have a codon that is AGT instead, but we've also altered this codon because now there's a C here that has to be read from this codon here, CGC. So our new sequence is changed from asparagine, glycine, threonine, serine to asparagine, serine, arginine, tyrosine, isoleucine, and all the amino acids downstream from this will also be changed. Could also have mutations in non-coding sequence. So here's a diagram of our eukaryotic gene, a nice little diagram for you to copy and memorize and be able to reproduce. Here's our start site of transcription. Here's an exon, here's an intron, here's another exon. Okay, here's our coding sequence. starts with an AUG and ends with the stop codon. Here's our consensus sequences for removal of our intron, GU, AG with an A in the middle. This is our cleavage and polyadenylation signal at in the 3' untranslated region. Here's our 5' untranslated region. Here's the core promoter with a Tata box, an enhancer, and a silencer. Everything you need for a eukaryotic gene. What happens if you mutate some of these sequences that are not in the coding region? This is just the coding region here. You have a lot of other nucleotides in this gene that could be mutate. What are the consequences of those? If you mutate the promoter region, the core promoter, you can have up mutations that increase transcription or down promoter mutations that decrease transcription. You can have splice junction mutations in eukaryotes. You can mutate the sequences here, but also other sequences that aid in the splicing machinery to find the GU and the AG and the A in the middle. And you can alter the 3' and 5' end by changing the nucleotide sequences, and that can alter the stability. It can alter the translatability. It can also alter where the messenger RNA is partitioned within the cytoplasm, either to the periphery near the edges or onto the endoplasmic reticulum. And you can certainly mutate enhancers and silencers. Any region of a gene you can mutate and have consequences. So this is some of the consequences of mutating nucleotides that are not within the coding regions. The promoter, regulatory elements, the 3' and 5' UTRs, splice site recognition, just what we just went over. Suppressor mutations. What are suppressor mutations? They're second site mutations that mitigate the effect of the first mutation. So you can have intragenic suppressors. That means mutations within the same gene. Here's a theoretical example. Here you have a transport channel that functions properly. You can have a mutation that alters its function, such as the mutation that occurs in cystic fibrosis. It's an ion transport channel, a chloride ion transport channel. It has a mutation in it. You could acquire a secondary mutation that mitigates the first mutation, and now this channel could be functional again. Intergenic suppressors occur when two proteins cooperate within the cytoplasm to perform a function together. So you can have a mutation in one protein component and have an inactive complex, but then you could have a compensatory mutation in a second protein that now makes this entire complex active again. So in summary, we have three big picture ideas: that mutations are the raw materials for evolutionary change, although they are usually deleterious, and that there are many varied and overlapping repair and coping mechanisms that evolved to deal with mutations. We just saw one of those, the intra and intergenic suppressors. Wild type is the most common genotype in a population. The forward mutation indicates that you've got a mutant, and a reverse means that, or revertant means that you've gone back to the wild type. Point mutations change a single nucleotide at a time. Insertions and deletions within the coding frame usually result in frame shifts if they are in multiples of other than 3 nucleotides. Frame shifts completely change the sequence of amino acids encoded by the gene after the shift. They often result in premature stop codons, which produce truncated and non-functional polypeptides. Mutations in the non-coding regions of genes also alter the production of gene products. And finally, compensatory suppressor mutations can be intragenic within the same gene or intergenic when there are interacting gene products.